Give you a brief status of islet transplantation and isolation technology. We will not go through the history of this. That is available online, open access in Cellar 4 if you want to view this nice review article that was recently published, but also the NIH consortium of multi-center trial, coordinating multi-center trials in North America and Europe, just completed 2 phase 3 trials in islet transplantation for the most severe cases of type 1 diabetes. So hopefully both for islet transplantation alone and for islet after kidney transplantation. So we're hopeful and confident that this will move towards the biological license application of what could be the first approved biologically active cell product in the United States. The procedure is already approved in other countries and continents. This is I cannot give you detail on the primary endpoint because the paper is not accepted yet, but here you can see at 2 years the insulin requirement and C-peptide generation from the transplanted islets with a very good safety profile. So today, insulin independence can be achieved in approximately 50% of the cases at 5 years, approaching the outcome of pancreas transplantation as an organ. But most importantly, 70–80% of these transplants have significant graft function at 5 years with normalization of hemoglobin A1C and resolution of impaired hypoglycemia awareness and no severe hypoglycemic episodes, which is the reason why these patients are candidate to transplantation. So now to quantum leaps that will be discussed also in this session, but what we need to do to make this procedure a routine procedure in diabetes is both optimizing the transplant site, but also avoiding the need of anti-rejection drugs that limit the applicability to the most severe cases of diabetes, and then the problem of supply, where we will generate enough cells to transplant the millions of people, and I'm not talking just the type 1, but all the type 2 diabetic patients that require insulin over time because they go towards beta cell exhaustion, and also, of course, cost efficiency. Our approach in Miami, just report because it's a very new development clinically, is bioengineering an endocrine pancreas in the abdominal cavity that would have the same supply of the pancreas draining to the portal system, but also where you could introduce other platform technologies to provide local immunomodulation, co-transplant mesenchymal stem cells or Tregs, or cells that could help establish tolerance in the local microenvironment, or immunoprotect with conformally coated semi-permeable membranes that could provide protection from the immune attack. We just started this IND approved FDA trial phase 1/2 of transplantation, and the first patient was transplanted last August. The technique is very simple: minimally invasive surgery, seeding a fully resorbable biologic scaffold that is comprised of the islet mixed with autologous plasma from the patient on blood and human recombinant thrombin. So you seed the islets with the plasma first, and then you add the recombinant thrombin through laparoscopic surgery, and then fold the omentum, providing then a new bed to revascularize the implant. And this is how it looks at the scanning electron microscope. The 3D scaffold, you see 2 islets here and the tip of an islet here. And this is the first patient transplanted, was on 31 units a day insulin before transplant, with high glucose glycemic variability at continuous glucose monitoring system records, and most importantly, severe hypoglycemic episodes that are the reason these patients become candidates. And after transplant, we were very surprised to see one of the best islet transplant result outcome ever. So now we're trying to understand the possible reason, but this was off insulin completely at a 75-month visit. This is the glycemic variability with glucose in the perfect range without any exogenous insulin. So we have now several centers across the various continents that are being trained in this procedure. The first one in Europe was transplanted a couple of weeks ago, and we cannot do the whole procedure in Miami alone because it's very expensive islet transplantation in the United States, but we have several centers from Australia, North America, Canada, Europe, and elsewhere that will join us testing this new site and technology for transplantation. So I'm thankful to all of our collaboration worldwide and the Cure Alliance for helping us bring in this new technology to open access as always. Thank you, and I will invite now Alan Colman to give the first.
Good morning, everybody, and thank you, Camillo, for the introduction. So I'm going to talk about the current progress in making beta-like cells from a pluripotent stem cell source as a means of solving the difficulty of procuring enough islets for islet transplantation. And I'll be giving you about 5 slides, so I won't be very long. And I'll start with why one would want to make beta cells from pluripotent stem cells. Well, there is an unmet clinical need for all type 1 and many type 2 diabetes patients, those who have to take insulin. Insulin is very good, but it doesn't, even with the best compliance, it's difficult to control blood glucose, and some people just find, even with insulin, very difficult to keep blood glucose within the requisite levels. Camillo referred to his pioneering work on making islets from cadaver-sourced people, and the proof of concept was established in 2000 by J. Shapiro in Edmonton of islet transplantation, and as he said, it's now improved so that you can get 50% survival, so 50% of the patients is still working well at 5 years out with the current immunosuppression regimes. But there's still a huge undersupply because there just aren't enough available cadavers with suitable islets. So there's a proof of concept, and one of the nice things about this is the transplantation technique is there's no need for physical integration of the transplanted cells as there would be if you were trying to repair a heart. I say this as a non-surgeon, it's a relatively facile transplantation into patients. But the problems, and I'll be alluding to these later, we'll need a very large cell dose of anything between 10 to the 8 and 10 to the 9 beta cells for a patient. And that's a lot of cells, and with that numbers come a lot of other problems. These cells must be allergenic for type 1 because it's immune rejection, and if you used autologous cells, even made from the same patient, say with the induced pluripotent stem cells as a starting material, they would be rejected. So you might as well start with allergenic because autologous wouldn't work. It's a very challenging, as you'll see, expensive differentiation, and some would say the healthcare economics will be contentious. I mean, it'll be very, very expensive, and the question is whether it'll be affordable, even if it can be done. So a little selected chronology. I mean, this all really started in humans with Jamie Thompson's reporting of human embryonic stem cell lines in 1998. In 2001, the paper on the production of insulin-positive cells made from human embryonic stem cells by Asadi et al., but it was something like 0.01% were insulin-positive, and they weren't beta-like cells, they just made insulin, but they certainly didn't make it in a glucose-responsive way. And at that stage, people got interested in, this was spontaneous, in trying to do directed differentiation, and I was actually chief scientific officer of an embryonic stem cell company in Singapore at the time, and we made this a major objective of trying to do recapitulation of the embryology involved in a dish to make these cells beta-like. We got some, but compared to one of our competitors, who I'll refer to heavily in a minute, we failed. It's a very difficult differentiation, and as you'll see, ViaCyte, a company based in San Diego and in Georgia, have succeeded pretty well. So from 2005 to 2008, there was pioneering work published by scientists at ViaCyte. And then the next publications I will want to refer to is 2014 with 2 papers which actually made glucose-responsive cells that were glucose-responsive in vitro. ViaCyte didn't achieve this, but that might not matter, as you'll see in a minute. So I'm just going to concentrate for the remainder of 5 minutes on these papers highlighted in red. So this is what people have been trying to achieve. You see on the top human pancreas development, a series of inductions from the inner cell mass going forward through to what's called stage 8 plus here, which is the mature beta cell. It's a process that takes nearly 9 months. It's a long process, and people have been trying to recapitulate this in a dish and have achieved it in about 5 weeks. So in 5 weeks you can do what the body does or the fetus does in 9 months. And it's a very a sort of complex procedure, and basically you try and recapitulate what is done in normal embryology and use the data that's been painfully assembled over the last 30, 40 years by developmental biologists, so that you can choose the right growth factors, tissues to enable this induction and this change to take place in vitro. So at the present time, ViaCyte, who I've mentioned as the pioneers in starting the process, they make cells which they would call endocrine precursor cells, and those are the cells they use actually for human transplantation at the moment. They're not insulin positive, but they become insulin positive in vivo. Whereas 2 groups in 2014 published quite similar, but there were significant differences in the papers on making in vitro glucose-responsive cells, which are much more mature than the ones ViaCyte have done. And what I'm going to do now is just compare the different preparations. So this is a comparison of the protocols and the citations involved. So ViaCyte are represented here by Kroon et al., and then the other 2 papers are Rizania et al., who are associated with company Betalogics, I think it's Janssen or J&J company, and Pagliuca et al., the lead investigator there is Doug Melton in Harvard, and they're associated with a company called SEMA. And I think you saw on my first slide a disclosure. I'm on the Science Advisory Board of SEMA, so you must take that into account. But I'm not here to try and badmouth anybody. I'm just trying to give what I hope is just an unbiased comparison of these various procedures. And I've broken it down into the cell type that is made. It's pancreatic progenitors with Kroon et al., and matured beta cells with the other 2 2014 publications. Matured means sort of mature, but they're not completely mature in either case. They're like beta cells, they have many of the features, but if you compare them head to head with primary beta cells, they're not quite the same. Now, one argument is, of course, that primary beta cells are taken under duress, either from dead people or from diseased people, and so we don't know that the beta cells that we call primary are representative of what we have in situ. But anyway, they're not quite the same. The production time and the number of steps, showing the complexity of the procedure, is the ViaCyte one, which makes the progenitors, takes 12 days and there's 4 steps. And the other 2 procedures take anything from 27 to 43 days, depending on which one, and have 7 or 6 steps, as shown here. The number of growth factors used, and when I say growth, it's not just growth factors, tissue modulators too, is 6 in Kroon and 11. That's a large number of things that have to be GMP compliant for a differentiation procedure. So there's a lot of growth factors involved in this transition. The percentage of insulin-positive cells is 0 in ViaCyte, and about 45 in Rizania, and about 33 in Pagliuca. The Rizania is based on immunohistology and is a sort of soft number, but That's what you're looking at. The other cells are alpha cells and unknown cells. So these are not pure preparations that are made. Scalable process, yes, in ViaCyte. Resenio say it's very difficult to make theirs a scalable procedure because there's an air-liquid interface step, which is difficult to scale up to the billions of cells that you need. Whereas the Pagliuca, they use three-dimensional 3D culture and relatively easy to get large numbers, billions of cells. Glucose responsiveness in vitro, you see the results there. And then when we go into the mouse models, you can see that the Krunital, the ViaCyte, takes 3 or 4 months for the cells to be producing insulin in mice, with recovery of fasting glucose levels in vivo, again 3 to 4 months. And this is because the cells you're introducing are not mature, and they mature in vivo. Now, that might be good, or it might be a problem. But as you see below, it's not such a problem that it wasn't an approved treatment by the FDA. So ViaCyte are now into people with these cells. They do it at 2 sites, one in San Diego and now in Canada, in Edmonton, are doing transplantations, phase 1, phase 2 trial, low numbers of these cells in the first safety trials. But you can see that the 2 later papers, the 2014 papers, are using cells which are already insulin positive and glucose responsive, and so consequently they act much faster when they go into mice. But those 2 publications have not yet matured into clinical applications. So my final slide is just what the remaining challenges are, and there are a lot of challenges. I mean, I'm not the best person, I think here probably Camillo is the best to talk about the best site for transplantation. The original islet transplantations were through the hepatic portal vein into the liver, but other sites are being considered and trialed at the moment, as he pointed out. Prevention of rejection, I told you these are all allogeneic cells, and what ViaCyte are doing, and probably what the other groups will be doing, are using devices, immunoisolation devices. In the case of ViaCyte, their site is just subcutaneous. They have a Theracyte device which they pack with cells and put under the skin, and the idea is they produce insulin but are not attacked by the immune system. Whether cytokines and things get into the devices remains to be seen and kill the cells, and I mean this is the purpose of the trials. But immunoisolation has the benefit that you don't have to worry about immunosuppression, has the great difficulty when you're using huge numbers of cells. Oxygenation is a real problem of your cell mass, and so these are issues that have to be explored. Then there's the longevity of the cells. How long will they last? Will they last as long as the person, or will you have to continually replace the devices or whatever over time? Or will the cells last but the functionality go away? And will they be functional anyway? We don't know that yet. Cell dose is an issue. Apparently the mouse is much more resistant to human insulin than the human is, and so we don't really know what the best cell dose is. It might be less than the numbers I gave you before, but it'll still be a large number. And there are safety concerns always related to pluripotent starting material. Again, immunoisolation helps you if some of these cells aren't differentiated properly and become tumorigenic, then they should be confined. But that remains to be seen. One of the problems with such large doses, if you don't contain them in this way, is it's very difficult to have an in vitro test of your batch to say that there aren't enough cells in that batch if you're putting in 10 to the 8 cells, to ensure that there aren't some rogue cells in there. It's a really difficult concern. And then there's the cost of goods issue, a very complex procedure, lots of factors, lots of media needs. How expensive would it be? Now, I think Camillo's best at talking about that. He knows the cost of the current procedures. I have concerns about the cost of goods and the health economics, because when people go through this, they really talk about the idea that you give a transplantation, a bit of post-operative supervision, and that's it. But if you have to go back time and time again and replace the device, it could be extremely expensive. So that all remains to be seen, and that's all I have to say. Thank you very much.
First of all, I would like to express my gratitude to the organizers for giving me the opportunity to be a speaker in this very exciting summit. As you know, it is of special interest finding a way to produce in vitro pancreatic beta cells to be used in type 1 diabetes therapies. However, in my view, cells generated in vitro are devoid of mature beta cell characteristics. So, finding new strategies to obtain mature beta cells remains as a major concern. Our approach in my lab started obtaining fibroblast cultures from type 1 diabetes skin biopsies, followed by its characterization using immunocytochemistry and microarrays analysis. Once confirmed the initial cell fibroblast signatures, we use a new chemical protocol only using small molecules for transdifferentiated skin fibroblasts into insulin-producing cells. The protocol details can be seen in our recent report. As a result, the chemical induction modified the cell morphology from fibroblasts to islet-like clusters. RT-PCR analysis of the transdifferentiated cell revealed the expression of insulin gene, glucagon, neurogenin, and PDX1. The qPCR analysis showed that in transdifferentiated cell, in transdifferentiated fibroblasts, glucagon and insulin were upregulated, and ASPN and MEOX, which are two genes related to fibroblasts, were both downregulated. And the microarrays analysis showed in a global way that our chemical protocols really activate the transcription of pancreatic genes, downregulate the expression of fibroblast transcripts, and thus not active embryonic cell markers. It's a transdifferentiation. In addition, glucagon was found in the 30% of the transdifferentiated fibroblasts by immunocytochemistry, and C-peptide production was confirmed by immunolyte analysis. In this transdifferentiated fibroblasts, G-binding revealed a normal karyotype. Methylation analysis showed no global change, and no teratoma formation were obtained in transplanted nude mice. However, the glucose challenge were negative, suggesting, as you know, the mature state of these transdifferentiated cells. Based in our previous results, we think that transplanting these cells in diabetic nude mice could involve their maturity. But we were wrong. The transplanted cells were unable of reverting diabetes and getting maturity. However, the transplanted cells prevent extremely preglycemic peaks, loss of weight, and in addition, human insulin was detected in the serum of 3 transplanted mice. Well, to improve our latest result, we began to work with a CRISPR-on system, which is a new tool for molecular editing described by Gilbert and colleagues. The CRISPR-on system consists of inactive DNA nuclease, Cas9, fused to an activation domains, BP160, and co-express RNA guides designed to hybridize a target sequence. In our case, the RNA guide was designed to recognize the promoter of insulin genes. These 3 elements combined can generate a DNA recognition complex that recognizes insulin gene in the transfected cells, activating the insulin gene by recruiting the transcriptional machinery. As you can see in this slide, we designed 4 RNA guides to generate a higher activation stimuli over the insulin gene. So, the cells were lipoaffected with these new CRISPR-on systems and sorted for further analysis. By RT-PCR analysis, our results revealed that the CRISPR-on system activates endogenous human insulin genes. In addition, by RT-qPCR, we observed a synergistic effect when all 4 RNA guides were used. In the epigenetic profile, the target promoter gene did not exhibit alteration in its methylation status, but rather altered levels of H3K9 acetylate histone, which is in proximal promoter regions, are known to be associated with transcriptional active genes. And our preliminary result by co-immunoprecipitation showed that the CRISPR-on system interacted with P300, which is a very low acetyltransferase protein. Lastly, we showed that this novel CRISPR-on system activates the insulin gene in a wide range of human cells in vitro, particularly in fibroblasts from type 1 diabetes patients. Now we have started to combine both strategies, chemical transdifferentiation and CRISPR-on method, hoping that the two approaches together can provide us an unprecedented level of precision for the control of cellular reprogramming until getting mature beta cells. Well, that's all. I'd like to express my gratitude to all the collaborators involved in this work, as well to the financial support. Thank you.
I'm going to talk about possibility to expand human and in general pancreatic islets in vitro. I'm actually a scientist working in the extracellular matrix, and I've been using extracellular matrix molecules to culture different kinds of cells, differentiate them from pluripotent stem cells, etc. And one of our attempts to see if we can find stem cells in platelets, in islets, we placed them on the islets on specific matrix proteins, specific laminins, and it turned out that with specific laminins we could actually make the islets grow. Now I have to disclose that I'm a co-founder and shareholder in several companies, the Nephrogenics Incorporated, which is developing a drug for diabetes complications, and we're in phase 3 clinical trial. And we also have founded a company called BioLamina, which makes these laminins, which I'm going to be talking about in my talk. I'm also a consultant for AstraZeneca. So the islet transplantation procedure for treatment of type 1 diabetes and now also moving into treatments of certain patients with type 2 diabetes has been quite successful. You can treat patients so that you can have good insulin production in the patients for up to 5 years. But it's still difficult to get this method to become robust, because it's difficult to get islets for these procedures. They are obtained from cadavers, as you heard before and many of you know about, but you need a lot of islets from cadavers to get a successful treatment. Camillo can correct me, but I think at least according to certain publications, in many cases one needs islets from up to 3 to 5 different cadavers to get enough for a successful treatment of a patient. So this is of course a big hurdle because it's difficult in the first place to get islets from one person and ship them around the globe for treatment, not to talk about getting islets from 3 to 5 patients or cadavers. And since year 2000, only about 500 transplantations have been performed. So the question is it possible to culture beta cells in vitro out of islets and get enough quantities for effective therapies by transplantation of islets or islet-derived beta cells? The generation of human embryonic stem cell-derived beta cells is very promising, as we heard from the first talk today. but there's still a long way to go before that becomes a routine method, I would think. The transplantation of beta cells, if they are pure, will require other cells possibly normally present in the islets, and is probably so that most of these islet beta cells, which are made from differentiated from embryonic stem cells, that they do have many of those cells actually in the preps. So we'll see how those transplantations are going to work. Now, when we were culturing these islets to see if we can find proliferating beta cells, we found some interesting results, and that really tell us that we can culture and expand the islets in vitro quite efficiently. Now, this work is based on work that we did already 9 years ago, together with a group in Dresden, at the Max Planck Institute in Dresden, Eckhard Lammert, and they were trying to study components, soluble components, secreted by endothelial cells in the islets. If you look at this picture here, you can see that this is a schematic picture. We have, of course, there are capillaries present in the islet, and the endothelial cells of these capillaries are sitting on a specific basement membrane, normal subendothelial basement membrane, and on the other side we have frequently the beta cell. Now, to make short story short, make the story short, it turned out that Lammert's group, they couldn't really find any soluble components that stimulated insulin secretion. However, they found out that if they had the whole matrix of the basement membrane located between the beta cell and the endothelial cells, they got stimulation of secretion in the system they were using. They were using insulinoma cells and so on. And we have been working a lot on the characterization of the basement membrane in this particular organelle, the islets, and they contain specific laminins called laminin 411 and 421, which are normally found in endothelial basement membranes, but also laminins 511 and 521, which are laminins which are ubiquitous, they're present in most basement membranes in the body, but they are developmentally regulated. And if we place the islets on these laminins, 411 and 511, we published in this paper in Developmental Cell that actually it was not the endothelial cells themselves or secreted factor, but the presence of these laminins, which was important in those experiments for secretion of insulin. Other laminins, such as the laminin which is in matrigel, which is a mouse tumor containing a lot of basement membrane proteins, the matrigel laminin doesn't have any effect. So if you keep the pancreatic islets in vitro on matrigel or the matrigel-derived laminin 1,1, they can just sit there in vitro. They can respond to glucose, but they don't expand in any way. If you put these islets in contrast on the laminins which are normally in the islets, these proteins laminin 421 or 521, they spread out quite rapidly, and you see a lot of proliferation of cells during this process. So if we, like here on the left-hand side, we have islets which have been sitting for 12 hours on laminin 111, and if we keep them there for almost 3 days, they stay there. They try to expand, but you don't see any expansion. And if you, in contrast, put them on laminin 5:1 and 4:1, which are normally in the islets, you can see that these islets are actually expanding considerably during the 2.5 days, which are shown in this picture. Now we have pictures showing expansion of islets for longer periods, up to 2 weeks, and you can see very extensive expansion. So something is happening. These laminins are somehow stimulating proliferation of the cells in the islets, a challenge that people have been dealing with for a long time, to try to see proliferation of beta cells and other cells in the islets. And we have been doing a thorough analysis of how islets behave on different substrates in vitro. Now here see a picture of many wells from a 96 well plate. They have been coated with bovine serum albumin, laminin 111, which is in mater gel. On the mater gel itself, on laminin 421, laminin 521 alone, and the laminin 421 and 521 together. These are the two laminins which are normally found in the basement membrane of the islets. And actually, if you compare the growth of islets in vitro on these different substrates, every picture here is you see here a single wells. In the presence of serum, actually, we see worse expansion of islets than in serum-free under the conditions we are using. This is a medium containing teaser R1, 5 millimolar glucose, I mean this serum part. But here we don't have any serum. It's the same conditions otherwise. And if we look at certain islets which are expanding here in the serum-free quite nicely, you can see that we have a lot of cells which are positive for expression of the C-peptide. You don't see in this auditorium here now any color for proliferating cells, the EDU expression, and it's difficult to see the DAPI staining, but I can assure you that we have good control of what's happening. And if you look in more detail at single islets, like in this case, I don't remember exactly how old these islets are. I think they're about 1 week in culture. You can see a lot of DAPI staining, a lot of staining for the C-peptide. These are not completely flat islets yet, so you don't see all the cells in here. There are also cells around, because these are isolated islets. They do contain probably fibroblasts and whatever other cells. You can see that they actually proliferate quite heavily. But if you look at the islets themselves, we can analyze every single cell by itself. This is using the operetta from Perkin-Elmer. We can make counts of how many percent of the cells in the islets are beta cells. They're about 30% in this particular islet, which is analyzed here, and about 3.4% of the beta cells are actually proliferating. So, we are doing a transcriptome analysis as we speak on what's happening in these islet cells when we place the islets on these particular laminins and compare with other conditions where cells don't proliferate. And we have very nice systems for imaging the cells in vitro, a specific marker, which is blue, taken up by the alpha cells, another compound taken up by beta cells, and then we are looking for new markers that can visualize the other cell types in the islets. So as a conclusion, the basement membrane, which is located in the islets between the endothelial cells of the capillaries and the beta cells primarily, contains specific laminins. And we have shown before that these two laminins actually seem to have some effect on insulin secretion in mouse insulinoma system. And we have been studying the adhesion of islets from mice, from monkeys, and now recently also human islets. They do adhere all to these laminins at 4:1, 5:1 in a completely chemically defined system, which is also xeno-free. And we can keep the islets viable for quite a long time, at least three weeks, and they respond nicely to glucose and stay healthy, as opposed to the islets that are placed, for example, on Matrigel, where they do not proliferate, and we see a lot of necrosis in the islets in 2 to 3 weeks. The mechanisms of these effects of laminins are not known yet, but we are doing a lot of work on characterization of the transcriptome in these islets during this treatment, and we hope to have those results soon. This we are doing with RNA sequencing and to get global transcriptome results. And I think it's a possibility for using these matrix proteins, these laminins, for keeping islets in good shape after their isolation at some place, and then during transport and during transplantation at a distant site somewhere else on the globe, possibly. So that's all I have to say about islets here, and I thank you for your attention.
Great, so now we can have a 15 minutes discussion, question from the audience. Just while you walk up, I'll just to comment on some of the comments you made before. Like, actually, the situation is a little better in islet transplantation, meaning that now we go for single donors. The case I presented is single donor of insulin completely. And since the first successful series of islet transplant, which by the way were not the Edmonton protocol, but were done in 1990, when I was in Pittsburgh, has been, now the progress is like to 70-80% function at 5 years, with several cases now going over 10 years insulin independence. The problem of cost is indeed quite dramatically, especially the difference, like in United States, an islet transplant can cost $250,000, $300,000. In Canada, 20,000. In Italy, 30. In China, 10. So depending on the country also, United States is becoming the most expensive place to do anything in clinical research, because you pay $42,000 just for the pancreas to arrive to the lab, even before you touch to try to extract islets. But that is another discussion. And but before asking my question, I'll open to the floor. You had a great, yes.
Does this work? Yes. I mean, I think there is a discussion between if you like, the two cultures, and I could refer to there was a paper earlier this year where people used blockage of the C28 co-stimulation thing, you know, anti-CD40, CTLA-4 to tolerize mice against human islet transfers, and that seemed to work to a point. But these strategies have been around for a long time, but seemingly have not been put into effect in humans for tolerization generally. So I have to think there are problems still in the conventional methods of tolerizing humans.
Sorry, where are you from? Can you say your name? And okay. So briefly, because it's not an immunology session, but this I think is one of the key issues that we listed in the opening, like that immunity and tolerance induction is a key new quantum leap that has to be done. There are major trials ongoing from Susan Ilstad and macrochimerism with now 19 patients completely off immunosuppression for up to 7 years post-transplant in the kidney transplant setting, and would be very interesting when we can use this technology in islet transplantation as well, because by replacing the immune system you obtain the double target of both inducing donor-specific tolerance but also eliminating the underlying autoimmune disease, so that you could think also doing this approach to restore self-tolerance and then promote beta cell regeneration or using patient-owned cells converted into insulin-producing cells. But are clearly very important strategy. We're involved also with the expanded Treg protocols, mesenchymal stem cell infusion that may be a component. I don't believe they will induce tolerance, but is a definitely a very important area of research and opportunity to synergize.
Yes, well, in those last two papers of 2014, when they looked at the insulin secretion after 20 millimolar glucose challenge, it was actually very similar to islet on a beta cell basis, very similar to the amount as produced by an islet cell. There was a variability for both, quite a wide variability, but statistically look quite the same. If you ask how many human embryonic stem cells or human-induced pluripotent stem cells you need to make one beta cell, it works out, I think, for both methods that you need 2 embryonic stem cells to make one beta cell. So if you're trying to make 5 times 10 to the 8th beta cells, you probably need a billion human embryonic stem cells. What we don't know is there's often where people have transplanted cells made from these type of sources into animals, there's been huge cell losses, 90% of cell losses immediately. So we don't know whether when they're immunoprotected or introduced in other ways, whether we can sort of suppress some of that immediate, which isn't immune related, it's just some sort of shock on introduction into humans. So we are talking about large numbers of cells. But I think that's possible. I don't think it's impossible to make so many cells, but it would be very expensive.
Melton said in the last ISCR in Stockholm that could be necessary 1 billion of insulin-producing cells to put in our patients.
Yes, and there is also a misconception on the number of beta cells that you actually need to reverse diabetes, because we start from the idea of what is needed to reverse diabetes with an islet transplant. But with an islet transplant, we're also using cadaveric, brain death organ donors that are not ideal donors, otherwise they would be still alive, so something bad happened to those islets, and then 60% of the cells that you infuse don't make it 48 hours post-transplant because of IBMIR, this instant blood-mediated inflammatory reaction. So that's one of the reasons we moved to the omentum and out of the bloodstream intravascular graft. But we published in New England Journal of Medicine a few years ago that if you use a healthy pancreas, in this case was an autograft from a soldier that was wounded in Afghanistan, and they were able to rescue half of the pancreas, 200,000 islets were sufficient to fully reverse diabetes, and that makes us think again, like, do you really need the 1 million islets or 700,000 islets if you have a very good source? So you may be surprised with a good source of healthy insulin-producing cells, the number may be much less.
Okay, I have a question. In regards to the last paper, the Bruns et al., that's 2015, that was comparing the mouse model to the rat model. And in your experience, what do you think would be the best model and you can see the difference between the mouse model and the rat model and how they responded to the doses that were in kidney capsule. So in your experience, what do you think would be the best model to study something like this, since there is so much differences? And you also mentioned how the mouse does not respond to the same, dose as it would be the human being or
yes, well, people say the rodent cells in general are more resistant to insulin effects than human insulin than human islets would be. People have used, of course, monkey models, but that's non-PC, understandably. and very, very expensive. So I think people will persevere with the mouse. And there are many different mouse models, and none of them will be perfect. I mean, they're helpful, but they're not perfect. None of them are going to really, they give you guidance, and you have to go into a human if you can get permission.
Hi, Mary Pat Moyer from Incel, and I'm wondering about the laminin work that's been done, and if you were going to make recommendations on doing manufacturing and using laminins, and where would the source be? What would be what are the better sources for the laminins? Are there already GMP manufactured laminins for this type of application? Because it looks like we can get to larger cell numbers and larger capabilities. Maybe we don't have to have cadaveric donors. There's a whole series of things that are relevant to providing that right environment for the cells. But if you can't get them to make them commercially viable, it'll be much more difficult if you're doing it, and of course increase the cost.
Well, it's not a problem to make them GMP, and they are actually, I think they are coming soon as GMP products, because there's a lot of groups and companies that are interested in these laminins for other purposes as well. So I don't think the source is a problem. The production capacity is not a problem for those laminins. And I'm wondering how it is because there's a loss of islets during these long transport times from isolation to the patient, if it would be helpful to plate them, the islets, on these kind of laminins, and then the laminins can be coated on films that are biodegradable, and you can then actually roll them up like cigars and transplant them probably like that under the skin or somewhere. So I don't think these are just minor technical problems.
Hi. I have two questions. One question is for the Alan and another one is for the Karl, basically. The question for the Alan is like, when you see the differentiation of these stem cells into the beta cells, do you see the differentiation into the another subtype of the islets also, like alpha and delta cells? At the same time, how does the microenvironment of the animals or the body is going to influence the differentiation into some different kind of the altogether cells? So that's the question for the Alan. And for the Karl, how does this integrins do affect on the different subtypes? Do there is effect on the alpha cells and delta cells also?
Well, to answer the first part of your question, when you do the differentiations, you get, as I said, a certain percentage of beta-like cells. There are also glucagon-producing cells, the alpha-like, and the somatostatin-producing. Generally, they tend to be lower in percentages than they are in the bona fide islets. I don't know what would happen to those after transplantation into a human. Certainly, when Biocide have put their precursors into mice, they get a mixture of all the cell types as they mature under the kidney capsule. But that's as far as I can tell you. But you get a mixture, whether you get the right mixture, and there are quite a lot of cells there which, frankly, we don't know what they are. They're not pluripotent cells, but they don't seem to be stained by any of the known antibodies against the other hormones.
Were you asking about the proliferation of different cell types? Right. Yeah. So you can see proliferation of all the different cell types, and if they are proliferating in the same ratio as in the original islets, we can't tell. There are clearly differences between different islets when it comes to proportion of beta cells versus alpha cells and other cell types. So we don't have that information, but all the cell types are actually proliferating.
Great, so we finished the hour, but if someone wants to remain and have a living room discussion, otherwise thank you all for coming, thank the speaker, and have a great meeting.